In this talk, we're going to learn about the central nervous system that arises from the ectoderm. After neural tube formation, the anterior portion of the neural tube starts forming the central nervous system. So, as part of the formation of central nervous system, there are three bulges called primary vesicles that are formed in the anterior portion of the neural tube. The anteriormost one, which forms the forebrain, is called as the prosencephalon, and it will form the cerebral hemispheres, and it is the portion that is shown in the blue box. The middle portion is called as the mesencephalon, and it forms the midbrain, which basically is parts of the cerebrum. And finally, the last part of it is called as rhombencephalon and forms the hindbrain, and this is where we will have the formation of cerebellum, pons, and medulla. Once the primary vesicles are formed, as development moves on, secondary vesicles are formed. These are usually formed by the time the posterior neural tube is complete. Now, in the case of the secondary vesicles, the prosencephalon, which was the forebrain vesicle, actually branches out to form two vesicles. One is called as the telencephalon and the other one is the diencephalon. And they form different parts of the cerebral hemispheres that are shown in the table that is to the right of the figure. The midbrain does not undergo any additional vesicle formation, and hence we have the mesencephalon the way it is. However, the rhombencephalon forms the metencephalon and the myelencephalon, so it also branches out into two secondary vesicles. Now, as shown in this table, it tells us the different parts of the brain that are formed based on their location of whether they are in the telencephalon or diencephalon or mesencephalon or metencephalon or myelencephalon. Now, when we look at the rhombencephalon region, the hindbrain of the rhombencephalon region will form a segmental pattern and even smaller compartments that is shown in the figure. These smaller compartments are called rhombomeres and have a numbering system associated with them, like R1, R2, etc. Now, each rhombomere expresses a unique combination of transcription factors. And what has been observed is that the cells of one rhombomere can mix freely within cells that are in that rhombomere, but it does not interact or mix with cells from another rhombomere. And thus, the rhombomere produces neurons that have different types of fates and different kind of functions. Once the neural tube gives rise to the primary and secondary vesicles, the cell populations that are on the wall of the neural tube start arranging themselves into different functional regions of the brain and the spinal cord. The neuroepithelial cells are multipotent and thus have the ability to differentiate into different types of nerve cells. They eventually differentiate to form the ventricular cells, which are also called as the ependymal cells. These line the neural tube and secrete cerebrospinal fluid. And the other set of cells that the neuroepithelial cells can form are radial glia. Radial glia are stem cells that show polarization along the apicobasal axis, and they have two main functions, and one of them is to act as neural stem cells, and the other main function is to provide a scaffold that allows the migration of other cells to give rise to the central nervous system. Radial glia are the neural stem cells that can give rise to progenitors that ultimately differentiate to form neurons and other cells like macroglial cells. Radial glia can undergo symmetric or asymmetric cell division, and the fate of the daughter cells depends on the plane of cell division. If the cell division occurs in an oblique plane, it normally results in an asymmetric cell division. The fate of the daughter cell in this regard depends on the centriole that it inherits. The daughter cells that inherit the older centriole become the stem cells, while the daughter cells with the new centriole begins the process of differentiation. While this is one mechanism that allows us to determine whether the daughter cells will be stem cells or undergo differentiation, there are other mechanisms that also aid in determining whether the radial glial cell will undergo symmetric or asymmetric cell division. The neuroepithelial progenitor cells can undergo differentiation to form the different cells in the central nervous system. Some of the main cells are the neurons and the glial cells. Now, among the glial cells, there are different types like the ependymal cells, the oligodendrocytes, the astrocytes, and the microglia. Neurons are the cells that conduct electric potentials and transform these electric impulses into signals that coordinate our functions. And thus, when we want to move our hands or move our feet, it is the electric impulses coming from our brains to the muscle that allows this movement to happen. When we look at the different parts of the neuron, the cell body that has the nucleus is called as the soma, and this is the portion that is shown in the blue box in the figure. The next part of the neuron are the dendrites. This is shown in this irregularly shaped structure in the figure, and they are hair-like appendages that pick up electric impulses. In some neurons, like the Purkinje neurons, we can see extensive branching of the dendrites, and in that case, they're called as dendritic arbors. The next part of the neuron is the axon, and that is the long structure that is shown in the blue box. The axon transmits electric impulses through the neuron. The last part is the synapse. The synapse is where you can have transmission of the electric impulses from one neuron to another neuron. And we can see in this figure there are 4 synaptic clefts. Now, in the case of transmission or transfer of electric impulses from one neuron to another neuron, neurotransmitters play an important role in this process. This process has been covered in cell biology, so please do have a look at it in case you want to refresh your memory. There are different types of glial cells, and we're going to start with the ependymal cells. The ependymal cells originate from neuroepithelial cells, and they form the lining of the brain ventricles as well as the central canal of the spinal cord. These cells have cilia, and one of their main functions is to secrete the cerebrospinal fluid. Oligodendrocytes are glial cells that insulate the axons of neurons that are present in the central nervous system by producing a sheath called as the myelin sheath. So when we look at the figure, we are able to see an oligodendrocyte that is secreting and producing the myelin sheath that is surrounding the axon of a neuron. In the other part of the figure that's in the larger blue box, we can see the myelin sheath that has already been laid by an oligodendrocyte. Now, interestingly, the thickness of the myelin sheath that is covering the axon is controlled by the axon and not by the oligodendrocyte. So the axon directs the oligodendrocyte on how thick the myelin sheath should be. The myelin sheath is a really important part of the neuron because it protects and preserves the nerves. In instances where demyelination occurs, it is usually accompanied with a disease. In the case of the peripheral nervous system, we have specialized cells that are similar to the oligodendrocytes that are called as the Schwann cells. So oligodendrocytes function in the central nervous system, while Schwann cells do the same function, which is to produce a myelin sheath around the axons. However, they function in the peripheral nervous system. The next type of glial cells are the astroglial cells that are also called as astrocytes. These are a group of cells that include radial glia and a variety of differentiated subtypes of astrocytes. They mainly function in the blood-brain barrier and responses towards inflammation in the central nervous system. They also support synapse homeostasis. The last class of glial cells are the microglial cells, and these are the immune cells of the central nervous system. They phagocytose dysfunctional neurons and glia and are able to get rid of them. They are small and motile, small in relation to neurons, which are much larger, motile in that they can move and that way they can sense and go to places where dysfunctional neurons and glia have to be cleared out from the system. Microglial cells originate from embryonic macrophage precursors, and hence they have an immune aspect to them. To summarize, we learned about different vesicles that are formed from the neural tube, which will give rise to the brain and other parts of the nervous system. We also learned about different cell types that are present in the central nervous system that included the stem cells as well as differentiated cells.